Hey, gang. I see some of you are here.
Hi.
Hi. How are you?
Good. How are you?
I'm good, thank you. When do you hop on your plane, Lily?
On Thursday.
Thursday. All right. Getting ready. Hey, Miss Claire. You taking over the garage seat? Very cool. That always looks like a comfortable spot to me. It's a nice day for it. Get your own little private spot. Did anybody else wake up and find their whole yard covered in frost? Because I did. It was pretty.
Nope.
It was very, very pretty this morning. It's great because, the back of my house faces due east. So, whoop, there's somebody. Yeah, it was because the sun comes straight at us in the morning. It's really cool. It was like the light was coming through all the little ice particles. It was all silver and sparkly. There we go. I think that's probably going to be our last one coming in. Or I don't know if Anthony not here yet. So I know that Darcy and Lewis are going to be out and Drew is out. So it's 10 o'clock. So we'll go ahead and start. So I hope you guys had an awesome Christmas and a happy new year. Hooray, hooray, we're here. I know it's been people around you probably dropping like crazy with COVID. That's been the case with us. Like everywhere we turn, somebody's got COVID. So we'll just do this online today. So we're starting our new unit and we're going to be talking about viruses and prokaryotes today because we're moving into our whole unit on microorganisms and how they evolved into plants. Hey, Colson. So here we are. Let's see. So let's first start with viruses because these are our most simplest of these organisms we're talking about. And the first thing I want you to know about viruses is that they are not living things. They actually contain genetic material, which all living things contain, but they're not technically alive. So you also can't kill them. So that's a hard thing to remember because when you're talking about stuff like COVID, like how do we kill COVID? Like what do we need to do to kill COVID on surfaces or whatever? You can't really kill it because it's not alive to begin with. There are certain things you can do that will make it inactive and ineffective. And that's kind of where we want to go. And I slip up all the time. I'll say the wrong thing, but we have to try and remember that viruses aren't alive. So what are they then? So virus is an infectious agent that can only replicate within a host organism. They are hijackers. They can infect a variety of living organisms, including bacteria, plants, and animals. So it's not just us, they can infect plants and bacteria. Viruses are so small that only a high-powered microscope is what you will be able to see them with. Technically, an electron microscope. Like we can't use any of our microscopes here and see a virus. They're even smaller than what even my best microscopes that I have can. There's Anthony. Perfect. And my last one. There he is. And they have a very, very simple structure. And their structure does vary depending upon the kind of virus you're talking about. So when a virus particle is independent from the host, meaning when it's out on its own, right, it consists of a viral genome, or basically it's genetic material contained within a protein that we call a capsid. And in some viruses, the protein shell is enclosed in another membrane that we call an envelope. And this is important to remember because when we talk about, if I put on hand sanitizer, will that kill any virus on my hands? Well, technically, no, it won't kill it because, like I said, it's not alive. But what it will do is it can destroy that protein shell or that envelope, and that can make the virus ineffective and basically disable it. And it works the same way with bacteria too. It basically breaks down the membranes. It's called denaturing a protein. And once that protein is broken down, the organism can't function the way it usually does. So viral genomes, meaning the genetic material in the virus, is very diverse. And it can be DNA or RNA. It can be single-stranded, double-stranded. It can be in a line or in a circle. It can vary in length and in number of DNA or RNA molecules. So there's a huge wide variety of what viral genome can look like. So if we talk about how viruses are discovered, we have to go all the way back to our basic understanding of what they are, and then we can look at how we got to figuring out what they are. So what they are, move this a little bit. So a viral particle is made of a nucleic acid, remember, like a DNA or RNA, and in some cases lipids. Remember, lipids are like fats and oils, right? And so what's important to remember here is that you've got some genetic material, some kind of protein, and maybe a fat. And that's really all that's going on there. They differ widely in terms of size, structure, and are so small that can only be seen with the electron microscope, like we said. Also want you to pay attention to this word here, viral particle. I've seen a lot of people with this COVID business going on saying things like virus cells and virus molecules and all that's incorrect. Don't say that. That means you don't understand what you're talking about, right? It's a viral particle. All right, and a typical virus has a core, or in the center, like RNA or DNA, surrounded by the protein, like we were saying. Some viruses, like influenza, so this is flu virus, have a membrane around that protein capsid. And the simplest viruses have only a few genes, whereas the most complex have hundreds of genes. So if we look here, this is like a very simple coronavirus. Oh, is this familiar? Never heard of that before? Maybe 1,000 times a day. So you've got your genetic material inside. So this is our genome, and this is an RNA. So that means that coronavirus is an RNA virus. And it shows here, you've got your membrane on the outside, and then you also have an envelope going on. And then you have these, and this nucleocapsid is basically how they're referring to the containment of the RNA, but it's not within a membrane like you would see in a typical eukaryotic cell. Remember, eukaryotic cells have membrane-bound nucleus. There's no nucleus here because this isn't even a cell. So these are spike proteins, okay? And these are going to be important because when we start talking about antigens in a little bit, you're going to want to remember these, okay? Lastly, we have these things called bacteriophages, and these are viruses that specifically infect bacteria. And these are really important because these are used a lot in research. So T4 is one that's used very often. And these look like aliens ships or something. They look like something NASA created, don't they? I mean, look at that thing. It's got like a head, collar, sheath. Oh, there he is again. We lost him and we got him back. So these are really good to know because we use them in research. Because if you want to know how viruses behave, or you want to do research on how they're transmitted, sometimes you can use your bacteria as your host in a lab. And then you can use these bacteriophages as your infectious agents. And that way you're not risking like living organisms that are more complex than bacteria. Like if your bacteria all die from a bacteriophage infection, it's probably not going to be ethically unsound, as opposed to if you had like a ton of mice or monkeys that you were unnecessarily infecting when you could have used bacteria instead, right? So they're really helpful for research purposes. So I'm going to scooch this down a little over here for a bit. So we go all the way back to 1892. So this is almost turn of the century, like Victorian era, when people were wearing like awesome clothes, my favorite clothes and furniture ever. I'm telling you, these people knew how to dress and decorate. All right. So in 1892, we had Dmitry Ivanovsky, and he's this 28-year-old Russian biologist. And he demonstrated that the cause of this particular plant disease called tobacco mosaic disease was found in a liquid that he could extract from the plants, but he couldn't pin down what was causing it. He just knew whatever it was in that liquid. And this is important because now we are getting a little bit closer to something that was causing a lot of problems. Like tobacco mosaic disease was causing some issues in their agricultural industry. So he knew it was in the liquids. Now we can keep looking. So now we get just a little bit later, just 5 years later, we get this Dutch dude. And his name is Martinus Beijerinck. I have a hard time with that one. I think it's Beijerinck. I'm not great with Dutch names, man. When we visited Amsterdam, everything ends in like Aken Paken. So we had a whole family joke when we would leave in the morning. We're like, who has the Bakken Paken? Like, who has the backpack? Everything there is crazy. Beijerinck. So anyway, he suggested that there were particles in the juice that were causing the disease. And he named them viruses, and he was using the Latin word for poison. So he's the guy who coined the term virus. And that's important because now he's famous for coming up with that word, and he'll always be the guy that's done it. So now we get all the way to 1935. So we've gotten several years down the road now, almost half a century later. We get this dude, Wendell Stanley. Okay, he's an American biochemist. And what he did is he was able to crystallize tobacco mosaic virus by taking that tobacco mosaic virus liquid and make it crystallize. And living organisms don't do like this. They don't crystallize. So he knew that whatever it was not alive. So now we can say that we know the infectious agent is in the liquid, it's a particle, and it's not alive. So this is the conclusion that biologists still recognize as being valid today, that a virus is not a living, not a living organism. It's a particle. Organisms like a loose term there, but it's a non-living particle made of proteins, nucleic acids, sometimes lipids. So now we have our favorite amoeba sisters, all right? And they're going to give us a good viral overview.
Chances are in your life,
Didn't mean to hit that. Sorry.
You've had the common cold before. It can cause sneezing and coughing and can make you feel awful. The worst thing about the common cold is that you can take medications to help with the symptoms, but you really can't treat the actual cold with antibiotics like you would a strep throat infection or an infected tooth. Why? Well, let's talk about what causes it: a virus. The common cold is caused by a virus. While all living things are made of one or more cells, see the modern cell theory, viruses are not cells. Viruses aren't technically classified as living things since they lack many characteristics of life, but there is some debate in calling them non-living as well. Medications like antibiotics attack bacteria like E. coli, which are living cells. Antifungal medications can be used on fungi like athlete's foot, which are made of living cells. Viruses are not bacteria or fungi. You can't classify a virus as a prokaryote or a eukaryote either because it's not a cell. So what is their structure like? First of all, viruses are much, much smaller than cells. You typically are going to need an electron microscope in order to see a virus. Viruses come in many shapes. Our favorite one to draw is the bacteriophage. It sounds very fancy, but it's a type of virus that tends to attack bacteria. Despite this being our favorite to draw, know that the shape of viruses vary greatly. One thing all viruses have in common is that they have some type of genetic material. This genetic material can be in the form of DNA or RNA. Viruses usually have some kind of protein coat, also known as a capsid. The capsid can protect the DNA or RNA genetic material. Viruses can also have other structures. Some viruses package special enzymes with them. Some viruses have an outer envelope. These additions can be useful to the virus in virus replication. One thing that makes virus replication or reproduction unique is that they can't replicate without a host. And it's their reproducing that tends to be a problem for its host, whether the host is you, in the case of the common cold, or an insect, plant, this bacterium. Viruses tend to be very specific for the host that they're going to infect, but anything that is alive can be a host because cells have the machinery that viruses need in order to replicate. We're going to talk about two types of viral replication cycles. The first one is the lytic cycle. In the lytic cycle, the virus, remember, very selective, attaches to a host cell. It often binds to a receptor that this cell has, which gives it access to attach. The virus can inject either its DNA or RNA, depending on what kind of genetic material it has, into the cell. Some types of viruses are actually taken inside the cell themselves. Now, you would think that the cell would notice viral DNA or RNA or an entire virus that has been taken in, but in many cases, it does not. It takes the genetic material from the virus and it starts following the instructions, which in this case is very bad because the instructions tell it to make copies of the virus. The cell uses its own resources to start building. It starts making so many copies of the virus that it can cause the cell membrane of the host to rupture, explode, lyse. So what happens is now that these new viral copies, they can get out of the cell and go and infect other cells. This is known as the lytic cycle. By the way, the lysing of the cell membrane is a very bad thing for the cell. The cell cannot survive without its cell membrane. The other cycle is called the lysogenic cycle. And I like to remember this is the longer word. So I like to think of this as the longer, sneaky cycle. Viruses that go through this cycle tend to do the same thing at the beginning. They inject their genetic material, but this time the genetic material stays hidden in the host genetic material. What happens is that when the host makes new cells, it replicates its own genetic material and the viral genetic material. Then those daughter cells make new cells, and they also replicate their own genetic material and the viral genetic material, and so on. This is the lysogenic cycle. That may not seem like a problem, but it really becomes one if the virus gets triggered to go into the lytic cycle from that point, because then all those cells that had the viral genetic material can start assembling viruses. And as far as what triggers it to go into the lytic cycle, it can vary. It could be a chemical trigger, for example, or a lack of food for the host. Going back to the common cold, a lot of times it has to run its course. Your immune system will typically take care of it, and medications can help with the symptoms so that they don't feel as bad. But unfortunately, some viruses are extremely complicated, like HIV, a virus that can lead to AIDS. Like most viruses, it is very specific. It binds to a CD4 glycoprotein that is found on the surface of specific cells, such as helper T cells. The problem about this particular target is that helper T cells play an important role in protecting the body and the immune system. Since HIV goes after these immune cells, it can make a person that is infected with HIV vulnerable to other infections. There are medications that have been developed to help keep HIV from replicating as quickly, and there continues to be research into looking into ways to keep the virus from attaching, because if the virus can't attach, it can't insert its genetic material. A problem that continues to be a challenge is that many viruses, including HIV, can mutate. So a treatment developed against a certain virus type may not work on a mutated form. So with our talk about viruses, you might wonder, is there anything positive about viruses? Well, some viruses can play a useful role in gene therapy. Definitely something to explore more. Also remember how we mentioned that viruses can go after other organisms, like insects, for example. A virus that targets pest insects could be an alternative to a chemical pesticide. One such example virus that you may want to explore is the nuclear polyhedrosis virus. Still, it's important to consider that whenever talking about any kind of pesticide, chemical or viral, there could be consequences in the ecosystem. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
There we go. You gotta love them. They're always good. She always sounds so excited to talk to. So now let's talk about bacteria for a bit. Bacteria, everywhere. So what are bacteria? So bacteria are prokaryotic. Do you remember what that means? That means they have no membrane-bound nucleus, right? So the prokaryotic cells are comprised of two different types of organisms, bacteria and archaea, right? So their genetic material isn't stored within a membrane-bound nucleus. Instead, it's stored in a nucleoid, is what they call it, and it floats around in the cell cytoplasm. Prokaryotic cells are normally smaller than eukaryotic cells, with a typical size range of 0.1 to 5 micrometers in diameter. That's itty bitty. Prokaryotes are made up of a single cell, though they can pair up or cluster to form mats. So the two prokaryote domains, bacteria and archaea, split from each other early in the evolution of life. There's that in the way again. Bacteria are very diverse, and they range from disease-causing pathogens to beneficial photosynthesizers and symbionts. So all bacteria, except for mycoplasmas, which we'll talk about later, possess a semi-rigid cell wall composed of peptidoglycan. Okay, peptidoglycan. Do you remember what plant cell walls are composed of? Does anybody remember what they're composed of? We talked about it before, but it's been a little while. So plant cell walls are composed of cellulose. And fungi, which we're going to be talking about next week, their cell walls are composed of something called chitin. So bacteria are different. They're composed of peptidoglycan. And then archaea are also diverse, but the difference is that none of them are pathogenic and they live in extreme environments. Okay, pathogenic, we're going to talk about this word a lot. Pathogen is a disease-causing agent, an organism like a virus or a bacteria or a fungus or whatever that will cause disease is a pathogen. So archaea are not pathogenic. So they don't cause infections and they can live in extreme environments. And when we're talking about extreme environments, we're talking like those deep, deep ocean vents that are really hot or that have a lot of sulfur, or just most organisms can't live in places like that. So we have different structures and functions, but lots of different structures when it comes to bacteria. And this is also how we categorize bacteria. Very often we categorize them based on their shape. So prokaryotes, so these are our cells with no membrane-bound nucleus, are probably the smallest living organisms, and they can range in size from 0.15 micrometers, which are mycoplasmas again, to, oh, come on, okay, Ozzy, I'll put you down. Sorry. To 0.25 micrometers. So an example of that one would be chlamydia, and then 0.45 micrometers. Another example would be Rickettsia, which these are just different types of bacteria, to about 2 micrometers. So they can go from like a quarter of a micrometer to 2 micrometers. So that's a good size range. So certain prokaryotes, such as bacteria, occur in different forms. And here's an example. All right, so if you have a sphere and it's singular, it's called a coccus. Together, it's called a cocci. These are fun. You guys are gonna like these. These are fun words. So spherical is a cocci or coccus. Rod-like is bacilli. Singular one would be a bacillus. You may have heard of lactobacillus. That's what helps make yogurt, actually. And then we have comma-shaped ones called vibrio. And then flexible wavy ones that we call spirochete or spirochete, some people say. Corkscrew, a spirillum. And then some prokaryotes have a variety of shapes and sizes, and those are said to be pleomorphic. And when we talked about Rickettsia a little bit ago and mycoplasmas, these are examples of things that can be pleomorphic. So you've got a coccus, a bacillus, a vibrio, a spirochete, a spirillum, and then these pleomorphic ones that just sort of meh. Did you have your hand up, Claire? I see a hand. Or is that? I can't hear you. No, maybe not. She's trying to hit a button. That's okay. You can always type it. I'll look for it. So also, when certain prokaryotes divide, they cling to each other, and it's in a certain pattern. So if you get 2, it's a diplococcus, which is funny because there's a dinosaur called Diplodocus, if you remember that. So it's diplococcus is a paracocci. Streptococcus, you ever had strep throat? This is what you had, a type of streptococcus. Tetracoccus is when they're in a little cube. So you can have this a pair, a chain, a cube. You can have a little grape-like cluster, that's staphylococcus. If you've ever heard of a staph infection, that's what we're talking about there. And then also we've got streptobacilli, and this is when they're in a long chain like this. The bacilli are in a long chain. So here's how we make yogurt. We use bacteria to make yogurt.
I'm sure you know that yogurt comes from milk, but did you know that bacteria are also a key ingredient? Yes, bacteria. But don't worry, these are good bacteria and are often called probiotics. Your gut has thousands of types of bacteria happily living inside, whose job it is to aid digestion. Yogurt is thought to be a good vehicle to bring more of these good bacteria in. So in this video, we're going to learn a bit more about how yogurt is made. First, we need milk. Before we start the next steps, all of our equipment needs to be sterilized to kill off any unwanted bacteria and other microorganisms. Now we are ready to start. The milk is heated to somewhere between 85 and 95 degrees Celsius for between 15 and 30 minutes. This is a process called pasteurization and is also done to kill off any bacteria and other microorganisms that are naturally found in the milk. The milk is then homogenized. This breaks down the fat droplets in milk to make them smaller so that they stay suspended in the yogurt, rather than sinking and making it all lumpy. So we've sterilized our equipment, then pasteurized and homogenized the milk. Now the mixture is cooled to 40 to 45 degrees Celsius, and we add our special yogurt making bacteria. The mixture needs to be cooled first so that the bacteria aren't killed by the high temperature. You might be wondering why on earth are we adding bacteria when we've gone to all the effort so far of killing all the bacteria. We're adding lactic acid bacteria, which are also used in cheese manufacturing. The two species used to make yogurt are lactobacilli and streptococci. But don't worry, you don't need to learn these names. Just remember that lactic acid bacteria are added. These lactic acid bacteria are known as the starter culture. The bacteria ferment the milk and change it into yogurt. Lactose is the main sugar in milk, and during fermentation, the bacteria turn the lactose into lactic acid. The bacteria also start to digest the milk proteins. Fermentation takes many hours, and during this time, the mixture is kept at the optimum temperature of 40 to 45 degrees Celsius, so that the bacteria can work its fastest. As fermentation produces lactic acid, type of acid, the pH of the milk drops to about pH 4.4. This is why natural yogurt has a sharp, tangy taste. As the pH levels drop, the milk solidifies to become raw yogurt. Once the pH drops to a low, the lactic acid bacteria that make the yogurt also stop working, so the yogurt doesn't become too solid. The pH drop also stops any bad bacteria growing. The solidification happens because the proteins begin to coagulate. Coagulation is when something which is liquid turns into a more solid state. So we've sterilized our equipment, then pasteurized and homogenized the milk. Add lactic acid bacteria so fermentation happens, and then coagulation. The mixture is now stirred and cooled to 5 degrees Celsius, and we've made natural unflavored yogurt. At this stage, we can add flavorings and fruit. Now it's ready to be packaged and sold. Delicious. Can you match the words to the definitions of the different stages of the yogurt making process?
Delicious, he says. And you guys know, if you pay any attention, I'm always drinking kombucha in the mornings because I love kombucha. And kombucha is another one of these things that has living probiotics. They call them probiotics because they're good living organisms that your body needs. It actually says on here, it said, there's a light, 9 billion probiotics. And if I turn around on the back, it says per bottle at the time of bottling. Now, why does it say at the time of bottling? Because, well, it changes with time. But it has a whole list of the different types of bacteria that are in here that are good bacteria. Bacillus coagulans, Aspergillosis, all kinds of good stuff. Lactobacillus, there it is again. So that's a really common one, lactobacillus. So let's talk about diseases now. Now I get to the fun stuff. Diseases caused by viruses and prokaryotes. All right, so infectious diseases are disorders caused by organisms such as bacteria, virus, fungi, or parasites. Many organisms live in and on our bodies, and they're normally harmless or even helpful. But under certain conditions, some organisms may cause disease. So some of these organisms are helpful. We want them. We don't want to kill them. Some of them are not. So in biology, a pathogen, and this comes from your Greek words, pathos and genus. So pathos in Greek means suffering or passion. We're talking passion here. We're not like, oh, I love you, passion. We're talking passion like you may have heard it if you're a churchy person. You may have heard like Christ's passion or that's Christ's suffering. That's where suffering, same word in Greek here. And genus, which you may also recognize as looking like the word Genesis, which of course is the production or the generation. There's another word that's similar, generation, the beginning of. So producer of suffering is a pathogen. Yep, causes disease. So it's in the oldest and broadest sense, it's any organism that can produce disease, and it can be referred to as an infectious agent, or you can call it a germ. So the term pathogen came to use in the 1880s. And typically, a term is used to describe an infectious microorganism, such as a virus, bacterium. Remember, bacterium is going to be your singular for a virus. Protozoan, which we'll talk about more next week. Prion, which we're going to talk about in a little bit today. Viroid, which is like, it's just the nucleic acid part. It's just the genetic material, and it's missing all the rest. And it usually infects lower life forms like bacteria or plants, not animals so much. And then fungus. So human pathogens, these are the things that can get us. Viruses, which you've heard of probably. Bacteria, you've heard of. Fungi, I know you've heard of. That's, you're probably thinking like mushrooms, but there's lots of other kinds. Protozoa, which we'll talk about more next week. Parasites, I know you've heard of, and then prions, which you've probably not heard of before, but I think prions are fascinating. We're going to talk about them a little bit. So these are the things that pathogens can do. They can destroy cells, they can damage cells, they can release toxins. Okay, what's a toxin? That's a really loosely defined word, but it has to do with, okay, I'm just checking that chat. Right. So it has to do with basically either a waste product or a something that is toxic or a chemical that's dangerous to a cell. So they can release dangerous chemicals, or they can prevent waste products from being removed from cells. And standard precautions, and this is capitalized because it's a thing, standard precautions for infection control. Okay, so what are things we do to keep ourselves from getting infected? Hand hygiene, washing your hands. Anytime you're going to touch food after you've touched body parts, eyes, nose, mouth, all these things, right? Hand hygiene, super important. That's probably the most important one of all. Personal protective equipment, which you probably didn't think much about until 2 years ago, and now everybody's thinking about PPE, as they call it in the hospital, personal protective equipment. Gloves, masks, and eyewear. This in the past would have been just like, maybe one day if I go into medicine or lab work. Now it's every day still, right? Respiratory hygiene, cough etiquette. They used to just say cover your cough. Now they tell you to wear a cough into your elbow, right? Is that way you're not getting it on your hands? Sharps safety, engineering and work practice protocols. Sharp safety. So if you've ever been at the doctor and had a shot, you probably noticed that on the wall or on the counter, they have like a little container that they put the syringe in, and then it sort of takes the needle down inside of a little box so that nobody gets poked. And this prevents bloodborne pathogens from getting passed from person to person by a needle stick. If you work in a hospital, for example, and you're giving a patient an IV or a shot, you could also get poked just as part of the process. And if you do get a needle stick, there's a whole thing you have to go through. Like there's a whole process you have to go through for making sure that you didn't catch something. You have to fill out paperwork, and it's kind of a big deal. So we have to be careful with sharps. Safe injection practices. So aseptic technique for parenteral medications. Okay, first, parenteral medications are anything that are not given by like mouth or not like suppositories, things that stick up the other end. So basically, if it doesn't go into your digestive system, then they're going to call it a parenteral medication. So this is shots, IVs, things like that. And aseptic would mean trying without infection or without sepsis, which is when your body gets infected and reacts to it. So trying to keep things clean. And then sterile instruments and devices. And then obviously this is really important if you're in medicine or dentistry or whatever. You don't want to be using dirty equipment. Like think back to the Civil War, like when they were just starting to use field medicine. If you ever been to one of the museums where they have like the old field medical bag tools, like they had these big saws and they would just go from person to person. If you got shot in the leg or whatever, they just cut your leg off with the same saw they used on the next guy, which is not sterile. So now we sterilize things, and typically we do that by autoclaving, which is heating it to a certain temperature for a certain amount of time, which prevents anything from anything that's living on the instrument from continuing to replicate or live, or if it's a virus, it denatures the proteins. So viral infections. So now we are at something very topical, aren't we? Viral infections. So we have two different types, lytic and lysogenic, which the amoeba sisters went over. So we'll just touch it briefly. So a lytic infection is when a virus enters a bacterial cell, or it can be any cell really, make copies of itself and then causes the cell to burst or lyse. Okay, it's called lyse when it bursts. And then lysogenic infections are different because these are the ones that take a long time. Hello, Alfred. So some bacteria viruses, and it can be other as well, but in this case, they're speaking about bacteriophage lambda because this is another one used in research a lot, can cause lysogenic infections. So this is in which a host cell is not immediately taken over. So instead, they're going to put their nucleic acid into the host cell's DNA, and it's going to replicate along with the host cell DNA, and it's not going to damage the host cell. So as that cell multiplies and reproduces and makes more daughter cells, all the daughter cells are now carrying the viral DNA. So that can get passed for generations. So it's different. So a viral infection is a proliferation, which means like a big explosion or growth of a harmful virus inside of your body. They cannot produce without a host. So, bacteria can grow on a surface. Viruses cannot reproduce. They can live or not live, exist on a surface, survive, maybe you can say. I don't know if you can say survive if something's not alive, but they cannot reproduce without a host. They need to hijack. So they infect the host, like we said, they hijack them, and they use their internal machinery to make more of them, more of their own DNA, more of themselves. And with an active viral infection, so say you have COVID, there's your example, you have a virus that makes copies of itself, and it bursts the host cell when it lyses the cell, and that kills it, and it sets all these newly formed viral particles free. So in other cases, you can also have viral particles that kind of bud off a cell, like they kind of grow off of the cell over a period of time, and then they kill the host cell. So in either way, your new viral particles are then free, and they go infect other cells. So symptoms of viral illness occur as a result of both the cell damage, the tissue destruction going on, and then also your body's response to it. And this has been in particular the problem with COVID. And the reason they know part of this is because they've looked at people who are on certain immunosuppressant drugs called biologics, like I'm on a biologic for lupus. And basically what that means is that, so like if you take a biologic that mediates your immune system, it kind of keeps your immune system from being overactive, because some people's immune systems are overactive. And people on those types of medications, if they do get a COVID infection, they tend to not be as symptomatic. Like they're not sick, like let's go to the hospital because I can't breathe sick. And that's because their immune system that response has been sort of dampened by the medication they're on. So we know that part of the issue with COVID is that our bodies are just really, really reacting to it. So unless you're on something like that, you could have a really big response and be very symptomatic. So certain viruses, like the ones that cause chickenpox and cold sores, can become what we call latent, which means they hang around. So you can have a cold sore and that erupts and then it heals. And then that virus, though, it stays in your body and it just kind of hangs out, doesn't do anything. But when there's certain triggers, and they can be environmental things like stress or sometimes sunlight, we don't even know what all the triggers are necessarily, but it causes that virus to sort of reactivate, and then it starts replicating again and you get new symptoms and it makes new copies of itself, but it never goes away. It just kind of lives there. And with chickenpox, like cold sore, that's what happens with cold sores. Like you get one and it goes away, and then you get one and it goes away. Chickenpox, when it comes back, it causes shingles. If you've ever heard of shingles, like if you have parents and grandparents, most of the grandparents are probably at the age where they get like a shingles shot. And basically shingles is like, it gets into your nervous system and it causes extreme pain. And it's the same virus that causes chickenpox. And if you had chickenpox as a kid, then you're susceptible to getting shingles as an adult. Most of you have probably never had chickenpox because there is a varicella vaccine that has been in wide use for probably over 20 years now. So even if you didn't have the vaccine, enough people have that it's not a thing. When I was a kid, everybody had chickenpox, and you actually wanted to get it when you were younger, because the older you got, like the harder it was for your body to fight and the worse you felt. So vaccines. So how do vaccines work? All right, here's a very important topic to understand how these work. So remember, a pathogen is any organism that causes disease in your body. Each pathogen is made up of these subparts, okay? Think back to that. Remember when I said, remember these little spike proteins? So think back to that graphic of the little coronavirus with the little spikes on the outside of it. So usually unique, these subparts are usually unique to that specific pathogen, and they cause the disease. And the disease it causes, sorry. So the subpart of a pathogen that causes the formation of antibodies, which is your body's way of recognizing and fighting a disease, we call those antigens. So antibodies is what your body produces in response to a pathogen's antigen, and they're important to your immune system. So you can consider antibodies as soldiers in your defense system. I like to think of antibodies as locks and antigens as keys. So I have a video for you in a minute that kind of shows you what I mean by that. So each antibody or soldier in our system is trained to recognize one specific antigen. We have thousands of different antibodies in our body. Some get passed from your mother at birth, others you just acquire because you come in contact with things in the environment and your body will produce them. So when your body is exposed to an antigen for the first time, it takes the immune system a little time to respond, and that's the time when you're sick. And then you get better when your body has fully responded. So once the antigen-specific antibodies are produced, they work with the rest of the immune system, they destroy the pathogen, stop the disease. So antibodies to one pathogen generally don't protect against another pathogen, except when two pathogens are very similar, like cousins. So once the body produces antibodies, its primary response to an antigen, it also creates antibody-producing memory cells, and these are very important. And they remain alive even after the pathogen is defeated by the antibodies. So if the body is exposed to the same pathogen more than once, the antibody response is much faster the next time. So basically, you got to picture this. Say you have a little COVID virus and it has this little key on it, and every key is specific, right? And your body comes into your body and your body sees it and it goes, uh-oh, okay, because you have these little like scout cells that are always out there, white blood cells, part of your immune system. They see it and they go, oh no, I see a key, I need the locksmiths. And it will call the locksmiths, and the locksmiths are going to be your antibody producing cells. And they're going to come and they're going to produce a lock that is specific to that key. And they will basically lock the key. And once they've locked the key, that particular pathogen is not going to be able to function within your body. And so if you've got a bunch of those coronaviruses all in your body, you're going to put out a bunch of locks, and they're going to lock all the keys that lock. Everything's going to get locked. And eventually your body will get over the infection. When that's done, your immune system is going to produce these little memory cells whose job it is to just kind of float around and be like, who do I recognize in this room? Oh, you, I recognize you. And I know who I need to defeat you. Hey, locksmiths, I need those locks for that guy. And all those locks come out and lock all those keys again, which is how you build immunity to something. So if a person's exposed to a dangerous pathogen in the future, their immune system is able to respond immediately. Now, I think this one's a little quicker than the others, but this is really good with antibodies, antigens. This is your lock and
Pathogens
keys.
are foreign infectious microbes that cause sickness and disease. The job of our immune system is to fight pathogens. White blood cells are able to recognize foreign pathogens by their surface molecules. The immune system constantly scans the body for foreign molecules. If foreign molecules are recognized, the immune system will launch an attack. Foreign molecules that trigger an immune response are called antigens. The surface of a pathogen is covered in foreign antigens. Antigens have very specific shapes, and different types of pathogens have different antigens on their surface. When the immune system detects antigens, white blood cells respond by producing antibodies. Antibodies are special Y-shaped proteins that are made with a specific shape to match the antigen detected. Antibodies are marking flags that latch onto antigens just like a lock and key. Pathogens that have been flagged by antibodies are then destroyed by special white blood cells. The antibodies you've produced to flag one type of pathogen would not be effective against another. For example, the antibodies that flag the measles virus would not be able to flag the chickenpox virus. This is because antibodies and antigens work like a lock and key. The shapes must match. As you fight different viruses throughout your lifetime, you develop the ability to produce different types of antibodies. This is how you gain immunity against certain pathogens. This is also what vaccines do. They trigger your white blood cells to produce antibodies so that you become immune to a particular pathogen. Now you might be wondering, why do I get a cold virus every year if my immune system has already produced antibodies? Or I've gotten the flu shot, so why did I still get the flu? Well, some viruses, like the ones that cause the common cold and the flu, have many different strains. These viruses mutate frequently and develop different antigens. If the antigens change, then you will need to produce different antibodies to fight it. The flu shot will help you to produce antibodies against a few different strains of the flu, but there's always the chance that a different strain could infect you.
So this has been the issue with COVID and the different variants. So we had the Delta variant, and then we have now the Omicron variant, right? Now, I don't know if you guys noticed, but I posted in our forum a really interesting article like the day after you guys left for Christmas break. And it was about Omicron and why it is so much more contagious than the other variants, how it's easily caught. And I posed some questions to you guys and I gave some background information. And I'm actually going to make it an extra credit opportunity that I will put out to you guys in this week's email. But yeah, you may want to go read that. I'll link it. But if you guys can answer some of the hypotheses that I have or come up with your own based on this concept here, you can get you some extra credit. We're going to run a little long, but I'm going to try to speed it up a bit. Okay, so here are your most common viral infections in humans. Okay, if you ever wondered what they were, here they are. So different parts of the body. For eyes, you can get herpes in your eyes. Skin infections, this one, varicella zoster. What's that? Chickenpox. Molluscum, rubella, measles, Coxsackie, Epstein-Barr. Some of these may sound familiar to you. Some of them may be like, never heard of that in my life. But yeah, here's your common cold: rhinoviruses, parainfluenza, SARS coronavirus. See that right there under pneumonia? It can cause, I actually have a friend who just got out of the hospital because she got like kind of post-COVID pneumonia. So she got lung issues from it. And that's what one of the things I'm talking about in the article I shared with you guys, that the initial, or what we would call wild type SARS coronavirus, the first one that we all, 19, that hit everybody without being a variant, specifically impacted lung tissue. Omicron looks to be hitting something else. You have a question, Miss Claire? Your hand went up again. No, I can't hear you. No, let's talk. Okay, well, you can always type it. So bacterial infections, let's talk about these really quickly. So bacterial diseases include any type of illness caused by bacteria, duh. So you got millions of bacteria that normally live on your skin, intestines, genitalia, all parts of your body, armpits, whatever. You want some of them there. They're good for you. Some of them not so much. So your vast majority of these do not cause any kind of problems. They're helpful and they're even necessary. Sometimes they call them good bacteria, normal flora, healthy bacteria. And then you got your harmful bacteria that can cause infections and disease. And these are ones that are pathogenic. So bacterial diseases occur when pathogenic bacteria get into the body and begin to reproduce and crowd out healthy bacteria or grow in tissues that are normally sterile, which don't have bacteria growing in them, even healthy kinds. And harmful bacteria can also emit these toxins or chemicals that are dangerous and damage the body. So what do antibiotics do? You ever had antibiotics? Probably have. So antibiotics are completely, there are different classes of antibiotics and they have completely different bacterial targets, or they act on the same target, but in a different place. So your main targets in bacteria are their cell walls or membranes. Break that down, they don't work anymore. The machineries that make their DNA or RNA, or the machineries that produce their proteins, which would be their ribosomes, because you guys know proteins are produced in your ribosomes. So if these targets are absent or different in the cells, these targets are absent or different in the cells of humans and other mammals, which means that antibiotics usually don't harm our cells, but are specific to bacteria. However, they can cause some unpleasant side effects. Well, why is that? because sometimes they kill the good bacteria. So you may notice, like, sometimes if you get bacteria, you can get other problems. If you have bacterial infection, you take an antibiotic for it, it fights that bacteria, but it also kills the good bacteria. So maybe you get diarrhea or a stomach ache or all kinds of other things that can happen because those other, the good stuff that's keeping you healthy gets killed as well. And that's not the point. So there's different types. They have narrow spectrum and broad spectrum. Your narrow spectrum antibiotics work against more specific types of bacteria, and your broad spectrum antibiotics work against a big range of bacteria. So this is like if you're fighting a war, do you want to use your sniper or your cannon? Your cannon will definitely hit the guy you want, but he's going to hit everybody else around him too. Your sniper is going to take out one guy. Your sniper would be your narrow spectrum, and your broad spectrum would be your cannon. So the problem is that sometimes it's really hard to diagnose which specific bacteria is causing an infection. So broad spectrum antibiotics are often prescribed. So doctors will just throw a broad spectrum at something. And if that's not working, then they may take a culture and try and find exactly what is causing the infection. A couple years ago, my friend's son was helping someone clean up their yard, and he chased a ball down a hill into a ditch. And it was a ditch full of water, and he was running barefoot. And he ran into the ditch, and he actually got stuck because he got impaled. His foot got impaled on a piece of rebar. There was a metal rebar in the ditch, like a, piece of metal rod. And his foot came right down on it, and it went through his foot, and he was stuck. Like, he couldn't get out of the ditch because he was stuck in there. And it was this muddy, nasty water. And my friend's a doctor, so she went over to get him, and she had to pull him off, which must have been very traumatic. And she tried to clean it up herself, and somebody was like, yeah, you better have a surgeon look at that. So it ended up, anyway, after some time, it got infected, and everything they were trying to use to kill whatever bacterial the infection wasn't working. So they cultured it and found out that the bacteria that was infecting him was actually one that grows on wet, green, leafy plants. Because guess what? He was in a muddy ditch full of gross water. So that's why none of the typical broad spectrum antibiotics were working. So your most common bacterial infections in humans, you've heard of some of these, I'm sure. Some of these are going to look familiar to you, like Staphylococcus. There's your staph infection that you can get. And then like maybe, you may have heard of Listeria before. You can get Listeria from like improperly stored lunch meats, actually. Salmonella, I'm sure you've heard. These are food poisonings. E. coli, all these things. So lastly, we're going to talk about prions, and I know we're running over, so I'm going to try to go fast, but these are so fascinating that they're worth talking about. So a prion is a disease or transmissible spongiform encephalopathy. Okay, encephalopathy, meaning this right here lets you know that it's attacking nervous system tissues. Okay, these are really rare, but they are progressive, which means they continue to get worse. Neurodegenerative means they break down, they degenerate your nervous system tissues that affect both humans and animals. They're distinguished by, they have these very, very long incubation periods, and they're characteristic of brain and nerve tissue structural damages and neuron loss and failure to induce inflammatory response. So they get into your body, and your body doesn't respond to it at all. There's no inflammatory, let's fight this. There's no immune system response to them. They just get in and break down your nervous system and your brain. And they break down the tissue in your brain. This spongiform means like the way it's supposed to look, it breaks it down. And they are abnormal pathogenic agents that are transmissible, which means you can get it from something. And what they do is they cause your normal proteins to fold wrong. Proteins that are in your brain and nervous system normally, those proteins fold wrong. Remember we talked about how you make this big amino acid chain and then it folds into a protein, causes the proteins to fold up the wrong way. So a lot of people think that prions may actually be like the predecessors to viruses because they aren't even as complex as a virus. They're like a part of the viral genome, really. So it's the folding of the proteins that cause the issues, and they are rapidly progressive once they really get a foothold. Like they have takes a long incubation period, but once they get a foothold, it just starts to degenerate quickly, and they're always fatal. So our animal prions are things like bovine spongiform encephalopathy, which we'll talk about in a minute, chronic wasting disease, scrapie, one specific to minks, one specific to cats, and one specific to ungulated refers to hoofed animals. Okay, this one right here, bovine spongiform encephalopathy, is mad cow disease. You may have heard of mad cow disease. And I have an interesting video on mad cow disease. So, and then in humans, we have Creutzfeldt-Jakob disease, and there's a variant of it. And then this one, which has a great name, Gerstmann-Straussler-Scheiker syndrome, fatal familial insomnia. And this one's interesting because it actually gets into the human genome, so it kind of gets passed generationally, but it's super rare. And Kuru, and I have a video on Kuru too. Kuru is kind of how our understanding of Kuru and bovine spongiform encephalopathy go together. Like, this is kind of how we figured out what's going on with prions. So mad cow disease comes from, well, one of the places it came from, and you'll find out in a minute here, is scrapie. So scrapie is a prion that lives in sheep. And cows are supposed to eat grass and grain and things like that. But when you grind up sheep bones and throw it in with their food just to have a little extra because it's cheap, and sheep brains and stuff get mixed in there, you can end up with mad cow disease. And I've got just 2 more quick videos, but they're worth it. Trust me, they're interesting.
If you were around in the 90s, and especially if you lived in the UK, you might remember all the panic about mad cow disease. It's a disorder that's killed thousands of cattle since the 80s and 90s, and because a version of it can also infect humans, a lot of people were really freaked out. Today, thanks to safer farming, it probably isn't anything you need to worry about. And even though scientists still don't know exactly how it works, we can say one thing for sure. Your steak is almost definitely probably not infected. Mad cow disease is the common name for a condition called bovine spongiform encephalopathy, or BSE. It's a progressive neurological disease that affects cows. And over time, it causes severe brain and nervous system damage, which eventually leads to trouble standing, walking, and changes in mood, like increased aggression and nervousness. The spongiform part of the name just means spongy, because the infection creates a bunch of holes in the cow's brain where its cells should be. The first 2 cases of BSE were identified in the UK in 1986, but it takes a really long time for symptoms to show up. So scientists think the first infections probably date back to the 70s. During the outbreak's peak in 1993, almost 1,000 new cattle were infected each week, but the number has gone down dramatically since then. BSE is caused by a bizarre self-replicating protein called a prion. Other pathogens like bacteria and viruses use DNA to make copies of themselves, but a prion is just a deformed version of a normal protein that's found in cell membranes. Sometimes those proteins can go rogue and get bent out of shape, but right now we don't totally understand how or why it happens. And when those proteins become prions, they can bind to other proteins like them and make them bend in the same way. And then those messed up prions corrupt even more proteins, and so on. Clumps of them collect and spread in the brain and nervous system, eventually causing brain damage. But that takes a while, so symptoms usually don't show up until years later. All of that is terrible, but how BSE got started is almost as horrifying. It probably happened because cattle were being fed ground up meat and bones from sheep and other cows. Ugh. There's another prion disease in sheep called scrapie, and it's possible that scrapie prions may have jumped to cows through their food and caused this whole mess. But it's also possible BSE just showed up when a random protein folded the wrong way. No matter how it started, the cattle feed only made things worse, because after cows died of BSE, they were ground up and fed to healthy cows, so the disease kept going. Unfortunately, a version of the disease can infect people too. The human version of BSE is called variant Creutzfeldt-Jakob disease, or VCJD, and it's also caused by bent prions. We don't know for sure that it comes from eating infected cattle, but since they're both prion diseases and both outbreaks happened around the same time, most scientists think that's the case. Like with mad cow disease, the symptoms of VCJD can take years to show up, but once they do, things move pretty quickly. Brain degeneration happens in just a few months, with symptoms like trembling, dementia, trouble walking, and eventually a coma. Since no cure exists yet, patients usually die within a year. Worldwide, there have been about 230 cases of VCJD, and about 180 of those were in the UK. The rest were mainly in Europe, and there have been only 4 cases in the United States, but they were all picked up overseas. The rate at which people have been getting infected has gone way down since the 90s, but scientists will keep studying it because there are other kinds of Creutzfeldt-Jakob disease not caused by cows. Before mad cow disease was a thing, we knew about CJD as a rare condition that could appear if a random protein went bad, usually through an inherited mutation or through medical procedures like transplants. Today, we keep the risk of variant CJD low by giving cows safer food and making sure no nervous system tissue gets into our beef. And even though the risk is small, you also can't donate blood in the US if you spent too much time in high-risk countries or got a blood transfusion in Europe, because there's a chance prions could be spread through blood too. Mainly, it's those new farming practices that have really helped get the disease under control. And in 2016, there were no new reports of BSE in cows in the UK for the first time since the outbreak started, which is both exciting and a relief. Also, because mad cow disease is transmitted through nervous system tissue, there's no evidence you can get it from milk or the meat used to make things like hamburgers and steak. So your roast beef is not out to get you, which is always good to know. Thanks for watching this episode of SciShow, and special thanks to Patreon patron Scott Sorrell for asking.
Thanks, Scott. So the thing to remember about mad cow disease in particular and how it got spread is that it was the nervous tissue, likely the nervous tissue of infected cows or infected sheep being fed to the cows that caused the problem. So this same sort of infection control when it comes to slaughterhouse practices is important with things like E. coli. This is why you can eat a steak rare, because it's inside of the meat, but a hamburger probably shouldn't be because it's ground up, which means there could be exterior portions mushed in, that things that touch something on the outside, and now it's all mashed up together. So that's the difference. So lastly, kuru. Okay, yep, that does say cannibalism. You're not seeing things. All right, kuru is a human prion, and it was passed through certain tribal traditions involving cannibalism. So this is about as interesting and as weird and as macabre as it gets, but it's helpful in understanding our
In the
issues with
1930s,
prions.
Australian prospectors went to Papua New Guinea in search of gold. There they encountered tribes that no one had seen before. One of these tribes was called the Foray. In the 1950s, scientists came to the area to study these people and made a disturbing discovery. Out of the total population of the Foray, numbering about 11,000, roughly 200 people died each year from an unknown disease. Patients at first had difficulty walking and speaking, then began to suffer tremors, and in the later stages of the disease, lost all muscle control. They couldn't get up, they couldn't feed, and inevitably, they would die. The Foray called this disease kuru, which means trembling, and it's the name by which it became known in medical literature. A curious thing about it was that almost all the patients were women and children. Scientists searched for years to uncover the cause of the mysterious affliction, and finally they found it: cannibalism. The Foray cooked and ate their dead, and they did it as an act of kindness. The Foray believed that it was much better for a body to be eaten by people who loved the deceased than by worms and insects. Women and children ate the brains of the dead, while men ate other body parts. The most interesting aspect of the disease, it turned out, was that the pathogen which causes it isn't a virus, a bacterium, a fungus, or a parasite. It's a type of infectious agent that's completely new to science, one that could survive even at boiling temperatures. This newfound pathogen was a protein called a prion. Prions are proteins often found on the surface of our cells, but if we eat infected meat, eventually the protein reaches the brain and its structure changes and becomes misfolded. The effect of the misfolded protein is to cause neurological damage and ultimately death. After this discovery was made, The foray stopped their human-eating ceremonies. Although it's difficult to be sure that kuru has completely disappeared due to the long incubation period of between 10 and 50 years, researchers think that the last kuru patient died in 2009. In 2012, the epidemic was officially considered to have ended.
So that's interesting because was an isolated group of people who didn't know anything about infectious disease and were doing what they thought was within their tradition was the respectful thing to do. And it was actually over time causing, much suffering. So lab 18, here's how it's going to go. Just so that you're going to check Teacheries and you're going to view the video that I've made for the lab. It's not available yet. It will most likely be available by Friday midday. That is because due to all this wonderful supply chain mess, some of our supplies are late. So today wouldn't have been a good day to run the lab anyway. And it's going to take at least 24 hours to fully run the lab once I've begun it, because we are letting, if you go and look at your lab, you'll see that we have to let some things incubate. And then you're going to have an answer sheet right there on in Teacheries, and you're going to want to download it, print it, complete it, and turn it in class. Just bring it to class next week. If something happens and I can't get the lab out until much later, I will extend that deadline. But you should be able to complete the answer sheet in 10 to 15 minutes or so after watching the video once we have it all together. And you can work on the answer sheet as a group. Does anybody have any questions for me, or are you guys good to go? Everybody good? I think y'all are good.
Yep, we're all good.
All good. Okay, I'm hoping we'll see everybody back next week. I don't have any reason to believe we won't. So, oh good, there's faces. Hey, Lil. So
Hi.
you enjoy your flight, sweetheart. And it's crazy in the airport, having just been there with my daddy. It's but you'll do fine. All right, you guys. So I will see you guys next week. And you have one little homework assignment from this lesson, which should be pretty easy to do. And you should be good. Catch you guys later, okay? Happy New Year. Bye.
Bye.
Bye.